get a protein really clean, we turn to protein chromatography. It uses columns filled with little beads called resin that have different properties that proteins interact with differently. If we flow a mix of proteins through these columns, then the proteins are going to interact differently with the resin, and then they're going to come out or elute from the column under different times or under different conditions, like if you increase the salt or you add a competitor. Basically, you have this problem. You have this mix of proteins and there's only one that you want. So how are you going to isolate just that one? You can do this with protein chromatography by taking advantage of the fact that different proteins have different properties. So your protein has properties that make it different from other proteins. But it'll have some of the same properties as other proteins, but some of the different properties. And so it might have one property that's the same with this guy, one property that's the same with this guy, but this guy has a different property than this guy. So basically, you can take advantage of the different properties like charge, size, special tags in order to isolate your protein based on those different properties. And how you take advantage of these different properties is by taking advantage of how these different properties make them interact differently with the different types of resin. So you can use resin that is going to interact differently with different charged proteins or interact differently with different size proteins. And so in this way, you can separate proteins based on their different properties using beads with different properties. We can do it either gravity flow, so we have some sort of bottle full of different resin. There are different types, so we can separate things based on charge or size, or there's like special affinity for something. Like for nickel affinity chromatography, we can put like a His tag on our protein and then use this to specifically capture it. So we can do that in these gravity flow formats, or we can do this using this machine, this FPLC, this ACTA machine, where the columns are going to basically, you have all these different columns of all these different types, and then it's got a bunch of tubing that's going to pump liquid and through the column. So it could be the liquid with your protein, or it can be just like buffers, so salt stabilized pH buffers. And basically, you can change the buffers in order to like elute proteins differentially based on how they're interacting with the resin. You can combine multiple steps in order to isolate just your protein, taking advantage of, so if one thing might have similar results with one type of column, like it might co-elute or come out with the same one protein, then it's not as likely to also come out in the other type of chromatography. And so you can build this workflow where you go from one type of column to another. A common workflow that I use when I'm trying to get a protein really, really pure, because I need my proteins really, really pure for the types of experiments I'm doing. So some people don't go through like all of these steps if they don't need the protein as pure, but I want my protein really, really super duper pure. So what I typically do is I recombinantly express the protein. So basically I stick the genetic instructions for making it into cells, in my case usually insect cells, get the cells to make it, break those cells open, and then what I'm going to do is I'm going to separate my protein. And I express my protein so that it has a little tag on the end. I'll talk more about these tags later, but this is like an affinity tag. And so then I can use affinity capture chromatography, where I use that tag. I use a resin, a little bead, one of these columns filled with these little beads that have something that's going to stick to that tag. Then when I flow my protein through, my protein's going to get stuck, everything else is going to flow up, and then I can collect my protein. Now once I can like push it off the column, once everything else flows through and I wash everything else off, my protein's still stuck on there, then I add a competitor. So something that mimics that thing that on my protein, the tag, I add like a tag mimic, and that pushes my protein off because it like competes for it. We put a ton of it in it. And then after that, I can cut off that tag, but now I don't have the tag, so I need to go to properties that are like natural for the protein. And so then I separate based on charge using ion exchange chromatography. So we can use cation exchange, or if your protein has more positive charge or negative, if your protein is more negatively charged, you can use anion exchange, but as we'll talk about, it's looking at like regions of your protein, so you can actually use like either in a lot of cases. We just have to, you have to be sure about like the pH and stuff. We'll get into it. So after that, I separate based on charge, but now I have things that maybe they non-specifically bound to the first column. Then they had a similar charge to my second column, but they're not as likely to also have the same size as my protein. So now I can separate things by size based on size exclusion chromatography, where the beads have pores with different sizes, and the bigger proteins they are actually going to travel faster because they can't go through all the little pores, whereas the smaller proteins, they can go through all the little pores. So the workflow basically for me is I go from the lysing, go break open the cells, and I do the affinity chromatography, then I do the ion exchange chromatography, and then I do the size exclusion chromatography. And along the way, I'll be taking samples with SDS Page, which I'll talk about a little bit, and you'll see that, so this is going to show you like all bands representing the sizes of all the proteins in the mixture, and as you go through the purification, you'll see the other bands, they disappear, and you're left with just your band of interest corresponding to your protein. And so then at the end, you have this one, hopefully nice band, and you're like, yay, protein. And now you can do the fun part and play with it. But before you can play with it, you gotta do all this chromatography stuff. So let's talk a little more about how it works.
So yeah, so there are those two types. So you can basically do a gravity flow, where you just let it drip based on gravity, and you have a little stopcock thing so you can control the rate of flow. Or you can do it with the ACTA, in which case you have control using the machine about how fast the liquid is flowing through. I much prefer using the ACTA. It is easier, and you don't have to do it in the cold room, but I actually use the column chromatography a lot for the affinity capture step, which is the first step that I'm going to talk about. And so I like to use it for the affinity capture step because typically at this point, I have a lysate, a cell lysate. So basically you break open a cell and then you spin out the, you centrifuge it, you ultracentrifuge it, so you spin it really, really fast. The membrane bits, and then the insoluble stuff is going to get pulled to the bottom, and then you're left with the water-soluble proteins. And assuming that you're studying a water-soluble protein, then your protein's going to be in this like supernatant in this liquid. But this liquid, it's also going to have a lot of other stuff, like a lot of other proteins, salts, different molecules, and it's going to be really goopy. And if you try to put this really goopy stuff through these little lines of the ACTA, it might not like you. And it'll go, the ACTA is not stupid. The ACTA is really, really smart. And so it has these sensors, so it's actually going to control the flow rate so that it doesn't exceed like the column pressure limits. And so it'll go really, really, really slow if you're putting a lysate through sometimes, at least with like insect cell lysates. With bacterial cell lysates, they tend to be a little thinner and have less of an issue. But either way, you have, I find it easier to do this step a lot of times in batch mode. Typically, I'll bind to the resin in like one of those little like big conical tube things on the like hot dog roller for like an hour. We call it like batch mode. And then I flow it through the column. So I use that for like the affinity step typically. Not always. Sometimes I do the his tag on the ACTA, but I tend to do it in the cold room, which I hate, but it's worth it for the good protein. But then later when I want to have more fine control for the ion exchange and for the size exclusion, when I want more control over the pressure and the buffer composition, so I can do like a gradient and that sort of thing, the ACTA is able to mix buffers so you can have like a salt gradient. Whereas when I'm doing the elution here, I typically just do it a step elution, so I just add the competitor at like a set concentration to push things off. But I'm getting ahead of myself because I'm talking about how we're actually doing these various techniques, but I haven't told you enough about the techniques themselves. So let's get into the nitty gritty of protein purification. So the main things that we can separate by are like affinity chromatography, and size exclusion chromatography and ion exchange chromatography. So ion exchange chromatography is actually like a form of affinity chromatography. But typically when we're talking about like, if we use the term like affinity chromatography, when we're talking about something that has like a specific feature about it, rather like something kind of special, more special than just having charge, because all proteins have like charge, at least at various pHs. But so this will be something specific. So you can be, if you're doing like a recombinant protein expression, it can be a tag that you put on the end of it. So it's a strep tag or a his tag or a GST tag. And then you have some sort of matching resin. And so then your resin is going to bind your tagged protein and everything else is going to come off. There are also other types of affinity capture that you can use natural things about the protein. So if you have like a double-stranded you have a DNA binding protein, you can use like a resin that has DNA bound to it. If your protein has some other properties, then there are other types of columns that you can use. There are columns where you can have like an antibody against your protein. There's columns that bind to, they have stuff that like binds to antibodies, and then you can kind of make a custom beads that have antibodies against your proteins, and then use this amino affinity to purify out your protein based on that. And that, to those types of things, you're not actually having to add the tag. You often cut this tag off, though. We'll get more into this. With ion exchange chromatography, then your protein is going to be binding based on charged regions of the protein. So it's not necessarily like you can have like a positive charge region of a protein and a negatively charged region of a protein. But typically you look at like what the overall charges of your protein and go with that based on whether, when you're choosing what type of resin to use, positive or negative. And then size exclusion, you're separating by size. Why I said that ion exchange chromatography was a form of affinity chromatography, where is because in both of these cases, you're having your protein stick to the column, and then you're washing other stuff off, and then you're eluting your column. So you're eluting your protein. So then you're pushing your protein off. So basically in these cases, your protein is getting stuck, and then you're pushing it off. The difference with size exclusion chromatography is that the protein's not actually interacting, like it's not getting stuck on the resin. It's not getting stuck on these beads. It's just traveling through the beads at different rates. And it's doing this because these beads have these pores, they have like these tunnels, and these tunnels have different sizes. And so you can use different resins with different like ranges of sizes depending on the size of the proteins that you want to separate. So, but in any case, there's going to be this range of sizes of tunnels in these beads. Why this matters is because if something's going through and there's a bunch of, if it's a little thing, it's going to see a lot of tunnels that it can fit into. So it's going to have to travel further. If it's a really big protein, then the small tunnels are still there, but it can't get through them. So it gets to go around them. So it travels a shorter distance. And so this way it's going to travel faster. It's going to come out sooner. And so you're able to separate them based on size. And we'll get much more into all of these things, but the key here is that your protein is not getting stuck. It's just traveling slower because it's traveling a longer distance basically through this. So with affinity, you're kind of like sticking it and then washing everything else off and unsticking it. And so that's the case with ion exchange as well. But with size exclusion, you're not sticking it, you're just taking advantage of the fact that they're going to have to travel different distances through it. So one way I like to think about this is with like a museum analogy. So say you have a group of people and you go to a museum. And so different people are going to find different exhibits more exciting than others. Some people aren't going to like any of the exhibits. I know my family would always hate going to a museum with me because I would stop and read like every single placard and they're just like wanting to go through. So if you were just to take us through a museum, and it's like a science museum, and so I'd get really captivated and stuck on the exhibits, and they'd keep going through. So then they'd come off the column like way before me, and I'd be stuck on there. And I wouldn't even come out if I didn't have to. And so, but then at closing time, the museum is going to like kick you out. And so in the analogy of this is when we're adding a competitor or we're like increasing the salt or that sort of thing, to basically, it's like someone that likes science even more than me came in, which would be like crazy, but then they're like pushing me out of the way. And so I can't stay at the exhibit and I have to come out. So in this way, we're coming out of the museum at different times. And in this case, because we're interacting differently, we have different affinities for the museum exhibits. With size exclusion chromatography, however, here it's more just like there are different, we're taking different paths through the museum. So if you, the little proteins have basically, they have tickets to more exhibits. And so they have to, and if you have to go through all the exhibits you have tickets for. And so these little proteins are going to have to go through a lot of exhibits. So it's going to take them longer, even if they're like walking the same pace and they're not like stopping at any exhibits, they're going to have to travel further than the big guys, which don't have tickets to the exhibits. And so they're not going to travel as far. So yeah, with size exclusion, your proteins aren't really finding the beads are really, really boring for the proteins. So it's like a really boring museum, like the walls are just like bare. And so they're all just traveling at the same rate. But if you have a bus full of people and they arrive at the museum at one time, and they all start walking, then and they're gonna like take a car when they get out of the museum, then the people that come out first are going to be in the first car. The people that come out later are going to be in the second car. So say there's like a car that comes every 10 minutes or whatever. So basically you can collect fractions as things are coming off of a column. You collect fractions and you can collect them into, like, if the actor uses this little fraction collector and they're going to come off of the column in these various fractions. And then what you're going to do is you're going to look to a chromatograph. And so this is basically, as the protein is going through, it's going to go through a detector that's going to detect proteins based on its UV absorbance. So proteins absorb UV light, and then they will show up when they're coming out, and you'll have this core, it'll tell you like this is going to be collecting fractions, and so then you can take those fractions that have your protein and then look and see if, like, collect those fractions, check that your protein's actually in them, and go from there. So when you're reading a chromatogram, so basically going up, so what this Y scale is going to be the absorption. So the UV absorption. So the higher this is, the more protein there is, although it can also get tricked by your buffer and by like nucleic acids, but like have really high absorbance. So like if your protein binds DNA or RNA, expect that the signal is going to be like erroneously high, or not erroneous, but kind of like delusionally high if you're thinking that it's all proteins. So typically what you're doing is you're going to equilibrate your column. So basically you make the column so that it is filled with the buffer that you're going to use. Then you load your sample, you wash the column. So basically you're going to basically wash off anything that's not stuck to the column. So this is when we're talking about like an affinity chromatography or ion exchange chromatography. And you're doing some sort of elution where you're changing the buffer. You can do this with a gradient elution or with a stepwise elution. And then your protein is going to come off the column. So this is when you're talking about affinity, or you're talking about ion exchange. When you're talking about size exclusion, however, here you use this isocratic elution. So this term always like confused me, but isocratic is basically you're not changing the buffer. So with size exclusion, you're using the same buffer, but everything is traveling through, because remember, you're not trying to like push anything off or anything. They're traveling through at different rates. So when you're looking at the UV absorption, this is just going to tell you like all like the quantity of protein type of thing and where the proteins are coming off. It's not telling you anything about what the protein is. And like, so SDS page can give us a better idea about what the protein probably is, because it's going to allow us to see the proteins, the size of the proteins that are in there. And the relative amounts of those sizes. So we talked yesterday about Western blots, which is where you like probe and see if there are specific proteins there. But if you use SDS page and just stain it with like COMASI, so you're staining for all proteins, basically the SDS page is going to denature the protein, so unfold them and send them traveling through this little gel. So unlike the purification where we're talking about like bigger volumes of protein that we actually want to like save and do stuff with SDS page, we're just talking about like a little sample of the protein. So we take a little bit of the fraction that came out and we can run it on the gel. The proteins are going to separate based on their size. Then we can stain it for all proteins, and they're going to, we'll see a band corresponding to the proteins that are in there. And you'll see that as the purification progresses, your band should be getting cleaner and cleaner, where only the protein that you want is going to be left at the end, hopefully. So for example, here's an example. So for example, for this protein that I study, argonaut or AGO, I express the protein in insect cells. Unfortunately for me, this protein is an RNA binding protein. So part of it is going to be bound to protein, to these small RNAs that are already like in the cell, so these endogenous RNAs. And then I can't use the protein because I need the protein to be RNA free. So that's just kind of useless for me. And I need this RNA free, which is a lot less than. But the reason why I'm telling you all this is because I can use this ion exchange in order to differentially separate the two, which is really, really cool. But I'll use the ion exchange later, but first I'm going to do the affinity chromatography. And so I actually express it with a sumo strep tag. So sumo is a fusion partner. It's this little protein that the cells really like making. So if I can stick this protein onto like the end of my protein, then it helps with like solubility and also expression. And I have more on that in another post. But so I have this sumo strep. And so then I can use strep tag purification. And it's actually a TEV sumo strep. So there's a TEV cleavage site that I can use to remove the tag. So I've cut off the tag. So it has a specific site for this protease that's going to allow me to cut off this tag. So I cut off the tag. You see that the protein band shifts in size. And I still have these contaminating bands, and now I have an extra band corresponding to TEV, as well as the bands corresponding to the tag that I cut off. And so now I need to separate my protein from whatever was already in here that just non-specifically bound to the strep column. I'll tell more about strep columns in a minute. And then also from this TEV and the tag that I cut off. And I also, with my protein, I have to separate the endogenously loaded from the RNA free. So I'm going to do this with cation exchange chromatography. So because my main purpose with this step is to separate the these proteins, and other proteins that are just like non-specific that are going to like wash off in other portions, I'm going to follow this up with size exclusion. But so at this step, I'm basically considering these two populations of my protein to be kind of like different proteins because they act a lot differently. And they act a lot differently with cation exchange chromatography because the negative charge of the RNA is actually making this protein less positively charged, so it's going to stick less to the column. So I'm actually using a stepwise elution here. So I have high salt, a lower high salt, and then a higher high salt. So this is an example of a stepwise elution. For other proteins, I often, for cation exchange chromatography, I use like a gradient elution. So I'll have a gradient where I'm increasing the salt. As you increase the salt, you're basically competing off your protein because the salt is going to be interacting with the salt is these charged ions. It's going to be interacting with the resin and competing for the spots on the resin. So your protein's going to get pushed off. And so it takes more salt to push off things that are more oppositely charged to the resin. So now you can see that this RNA free fraction, which is the one I want, I purified out my protein, but it still purifies with these other things. If I had done a gradient, I might have been able to purify out those things a little better. But this is like this tried and true protocol that we use, and it helps really because it's really good at separating these two fractions of argonaut. And then I can use size exclusion to further purify this one because there's this big difference in size between this product I want and these other things that I don't want. So for this, I use size exclusion, which we also call gel filtration. And then you can see now that I'm able to purify away most of the contaminants. So now let's talk more in detail about these different techniques. So as I said, with affinity chromatography, we're separating based on something that, so as I said, like with affinity chromatography, we're separating based on something that's like specific about our protein. And often what we're doing is we're separating based, at least for this like affinity capture step, we're basically purifying it based on something really, really unique about our protein. And often when we're doing a recombinant protein expression, so basically we're sticking the instructions for making the protein into cells and telling them to make them for us. Because we're sticking in these instructions, we can tell the cells to put a little extra onto our protein. So we have the instructions for our protein, and then we have the instructions for like a tag and possibly a fusion partner, like the sumo or the GST or MVP or something. But this tag is going to allow us to use affinity chromatography that has specific binding for that tag. And so the the concept might seem kind of confusing, like, wait, how you just stick something on a protein? But really, it's just a combination of amino acids that is going to bind specifically to the resin. So basically, a couple of common examples that I've used are his tag and strep tag. So the his tag, you have like 6 to 8 histidines in a row. What this is going to do is it's going to act as like a chelator. So it's going to bind, a chelator is something that like bites down on metal. So it's going to bind to metal. And if you put nickel, you coat your resin with nickel, then it's going to bind to the nickel on the column. And then you can then flow, you can then basically flow your protein through. Your protein's going to get stuck. Other things are not. And then you can push it off using imidazole, which looks like histidine, but it's not. It's just this part. Because with your protein, so a lot of proteins have histidine because it's one of the amino acids. But in the way you can get specificity is by making it so that you have that long string of histidines. Because you have that long string, this is really making it good for chelating, where you're able to have multiple things like acting together to kind of bite down on it. And so other proteins are less likely to have like a string of histidines like this. When you add imidazole can also bind to the nickel, but because you don't have that effect of having a bunch combined, you have to add a ton of it. And so you can use imidazole as a competitor, and you really flood it with this imidazole. So you use some of the imidazole like in your buffers, even when you're just doing binding and stuff, to prevent the really non-specific binding. And then you can either like do a gradient where you increase the histidine gradually if you're using like an ACTA, or you can just do a stepwise. Sometimes you can try different final concentrations of imidazole in order to get your protein off. A good thing about the his tag, it's really short, and you can sometimes leave it on if you don't have like a strep tag, or if you don't have a fusion partner or anything. But with affinity tags, we often design them so that you can take them off if you want, by having a protease recognition sequence for a really, really specific protease or protein cutter, like TEV, or HRC protease, which is like commercially like precision protease. But yeah. Oh, one last thing before I move on, with the his tag, once you push this off, you can also have some leaching, where basically you have some of the nickel that's going to come off of the column. This can be a problem because you have a his tag on your protein and you have this free nickel, and so your protein can kind of like aggregate around this nickel, this free nickel, and so your protein can like aggregate or clump up. And so one thing that you can do to prevent this is like you can add to your elution. So you don't want to have, so EDTA is a chelator. So we talked about how histidine can act as a chelator and kind of like clamp down. Well, EDTA is able to basically do that like super duper well. It can clamp down on metals and hide them. So you don't want to use EDTA in your buffers if you are doing nickel affinity chromatography, at least in the beginning steps. So if you need to be careful too, if you're using a commercial protease inhibitor, make sure that it doesn't have EDTA. So you have to add a protease inhibitor, so to prevent like your protein getting degraded when you lyse the cells. But if it has EDTA, then it's going to interfere with the binding of your tag. But once you've done the column, then you don't have to worry about having EDTA. In fact, as long as you don't have like a metal binding protein that you're trying to purify. But if you at this point now you want to hide the extra free nickel so that the protein can't bind to it and aggregate up. So you can add, typically I add it to my elutions to like a final concentration of about 0.1 millimolar, and this will chelate any excess nickel. So if you're doing this you can even set it up if you're doing it in an ACTA, you can like put it in the fraction collector so that it'll come out right into the EDTA to the final concentration that you want. Sorry, that was kind of more technical than I meant to go into for these posts, but we're talking about this in lab meeting yesterday, where someone was having some trouble with an expression and some aggregation, and that was a topic that came up. So I tend to use a his tag when I'm working with bacterial cells. And the strep tag, when I'm working with insect cells, there tends to be less non-specific products. So bacteria have a lot of like things that are going to non-specifically bind to the resin because they have like biotin stuff and that sort of thing. So a strep tag is going to give you a lot of artifacts, and then human proteins can have a lot of his things. So those will give you artifacts with a his tag. So this is why I tend to use a his tag and a strep tag. Okay, so now strep tag. So strep tag I tend to use with the insect cells. So strep tag is going to basically bind to the streptactin resin, which is a streptavadin mimic in the same place as biotin and desthiobiotin. So there's this like biotin streptavadin like super duper strong interaction in nature. And it's so strong that like it wouldn't be good for us to use because we can never get our protein off. But there are these mimic forms that have less affinity, but still good affinity, and that allow us to take advantage of this in order to bind to a protein. So the strep tag is just an extra string of amino acid letters, but in this case, we have different letters. So with the his tag, it was just his. With the strep tag, we have these different letters and we have the sequence. And with the twin strep tag, which is what I use, there's actually two of these with then like a flexible linker in between. Well, so this is going to bind to the special beads, the streptactin beads, and then basically you can then compete it off with the desthiobiotin. And so your tag is going to bind, your tag is kind of like a biotin mimic, and then you put in another biotin mimic in order to push it off. We use this desthiobiotin, which is like, it's missing this part, this thio without the sulfury part, because it's easier to displace. So the biotin is going to, even though you have this like mutated streptavadin protein, it's still, biotin will still bind really, really strongly. So it's harder to get it off. And so we want to be able to reuse this resin. And so biotin would compete off our protein well, but then we couldn't compete it off of the column in order to then regenerate the column. And so by using desthiobiotin, then we're able to like wash out the desthiobiotin and get it off and be able to use this resin again. So yeah, so these resins can be used a lot. They're pretty awesome. Okay, but if you're having trouble, like you want to check, there's like this dye, like this HABA dye thing that you can use to check if your resin is still like good. It turns red. It's really cool. Anyway, more on that in my posts on the strep tag purification. Okay, so now we've used that special feature and now we've cut it off. So with some purposes, you don't need to cut off the tag, but if you want to make sure that your protein is like more natural, then you typically cut off this tag and make sure this tag's not interfering, especially if it's a fusion partner tag and not just like a short little his tag or an HA tag or a flag tag or something like that. And so with the, and the other thing, so those are just two examples of affinity chromatography. There's also like, you can use like amylose resin with like GST and like compete it off with maltose. There's like columns with things like DNA bound to it, things like antibodies, various different types of affinity chromatography. These are just the two that we tend to use a lot. Okay, now let's talk about ion exchange chromatography. So ion exchange chromatography is going to take advantage of the fact that proteins have different charges. And so the charge is going to depend on the pH. So there are some amino acids, some protein letters that are going to, sometimes they can be negatively charged. And then there are other ones that sometimes that are typically positively charged. And so proteins have this mixture of positive and negative, and it depends on the pH as well. So you want to make sure that your protein is like going to be charged where you want it. And so look at the post on the like protein charge and stuff if you want to know more about that. But there's this point, this isoelectric point, which is the point at which the protein is net neutral overall. So if you're higher than the isoelectric point, you are going to be more negatively charged, and then if you're below it, you're going to be more positively charged. And basically you can look up with tools like prop-param, the PI of your protein, this predicted PI. But what's happening really with ion exchange is that it's happening in like regions of a protein. So often you'll see people do like a cation exchange and an anion exchange, or that sort of thing, because you can take advantage. It might bind better to one of them, but it can also bind to the other. Sometimes you have problems where it's not binding to either, then you might need to look into the salt or use some sort of like hydrophobic column or that sort of thing. More on that in another post. I don't want to get into that here. But the point is that basically you want to use a resin that is going to be oppositely charged to your protein. And so if your protein's more positively charged, so the pH that you're using, you're going to, we call positive charge things cations, we call negative charge things anions. So with a cation exchange, the thing that's being exchanged is your protein is binding to the, so basically this resin, you don't just have like a bunch of negative charges like resin just like chilling. Instead, it's going to be coated with the counter ion. So it's going to be coated with the positively charged part of a salt. So basically you'll have this negative resin, and if you have, say, like a sodium chloride buffer, then the sodiums are going to be hanging out onto these beads. But you start with a low salt. So there's not that much, your protein is able to basically outcompete the salt and stick to the column. And then what you can do is as you increase the salt, then the sodium is going to come back and push out your protein. And so the more salt you have, and then it's going to compete more and more. The stronger the protein is bound, the more salt it's going to take to elute it off, to compete it off, to push it off. And so because you're exchanging your positively charged protein for the positively charged salt, you're exchanging the positively charged thing. So this is a cation exchange, the negatively charged resin. With an anion exchange, you have a negatively charged protein, and what you're competing here is the negative, negative part of the salt. So a salt is the positive part and the negative part that combine make a neutral thing. So you have a sodium positive, chloride negative. So here would be like the chloride that would be competing off your protein. So basically you can then increase the salt and make a salt gradient. And then the least opposite charged protein is going to come off for elute first, and then the most oppositely charged protein is going to come off last. You can also do a stepwise dilution, as I see with my ego purification. So this was taking advantage of something natural about the protein, because all proteins have a charge, at least at some pH, and then you can take advantage of this. And so, but remember this is like the net charge overall that we're talking about. So you still have some stretches of positive and some stretches of negative in all of these cases, even at your neutral point. But you don't want to be like at your PI typically, because it tends to be the least soluble, because water likes charged things. Okay, so now you've done this ion exchange chromatography. Oh, and I should mention that because we need to start at a low salt, because if we start at a high salt, then our protein is not going to like stick to this. And so if your affinity chromatography step is in like a high salt concentration, you're going to want to dilute it before you do this, but also be careful that if you dilute it too far, your protein might precipitate out, because proteins like to have some of that salt to help keep them soluble as well. So you might have to experiment a little to find what you want. But basically, once you come off the affinity chromatography, you need to make sure that you're going to dilute your protein to the concentration that you want to start at when you're doing the solution, because if you start at a high salt, then basically you already have your competitor there and your protein's not even going to stick in the first place, which you don't want. So now you've pushed your protein off of the ion exchange chromatography. And so now you have your protein, and it is going to have a bunch of salt with it probably. And so you might want to get rid of that salt. And one way you can get rid of the salt is with the dialysis, where basically you stick it in a membrane and then you like stick it in this bucket and like a bath of like liquid buffer that's like the buffer that you want, but it doesn't have the salt, and then the salt is going to diffuse out. It's kind of a pain in the butt, and it's not going to be purifying your protein any further. It's just removing the salt. You can also do size exclusion chromatography, and it has the benefit of, in addition to getting rid of that salt that you don't want and anything else, it's actually going to separate out proteins as well. And so typically what we do is then we take it to a size exclusion chromatography, and this is going to serve as kind of like a purifying step. So even if your protein is really, really pure after this, we typically go to size exclusion chromatography, both in order to get it even purer and to get rid of the salt and get it in whatever buffer that we want. So basically we fill the pores with when we equilibrate the column, we fill it with the buffer that we want. Then when our protein is coming through, it's going to come out with the buffer that we want. But in order to get this to happen, what's going to happen is we need to concentrate our protein. So when we're doing the size exclusion, we're actually going to be injecting our protein into this injection port. So with other methods, we could use like a sample pump, or it's going to like suck it up and put it on. Or if we're doing it in gravity flow, we just like pour it in and that sort of thing. But here we need to have a small volume. And the reason why we need a small volume is if we think back to like our museum analogy. If you have a bunch of people showing up at the museum at different times, even if they're traveling through at different rates based on their size. So basically, if the person that came in, like if you had a person with a lot of tickets, so they had to go through a lot of exhibits, so like a small protein, if it comes in way before a protein that is, if it comes in like way before a protein that is bigger, so a protein that would like have less tickets, less exhibits or whatever, so that one's going to travel faster, or it's gonna, but it's gonna come out at the same time. So basically they both be on the same car leaving the museum, which isn't what we want. So we need to minimize the volume that we add in order that they all like enter the column at the same time. But once it comes, so we're going to need to concentrate it. So basically we take the fractions that contain the protein, and then which we tell based on the UV absorption and the SDS page. And then we use these centrifugal filters on these like spin concentrators in order to concentrate down our protein. And then we can inject it into the machine, and then the machine is going to control it going through the column. More on this in my post on size exclusion. And so you can also think of size exclusion as kind of like these tunnels where you have cars of different sizes, and they're going to have to travel different distances based on their sizes. So the bigger things are going to travel faster, so they're going to come off the column sooner, and then the smaller things are going to come off later. And so this is like opposite of what you would expect if you were doing an SDS page gel, where basically the bigger things are going to travel slower. Here, the bigger things are going to travel faster. So when we look at one of these chromatographs, so basically first you want to equilibrate the column to fill it with the buffer that you want, because your protein is going to be coming off at this buffer. So the things in the buffer, like the salts and these little things, they're going to travel really, really slow. They're like going to be like the slowest things moving through the column. So basically whatever your protein is going to come out with whatever was in the column, but it's going to also take the column a long time to equilibrate. Then, so you equilibrate the column with like a couple column volumes. So like the volume of the whole volume that the column holds, you like double that. So now when you flow your protein through, so you inject your protein into the loop, it goes into the loop and then into the column, but it's not going to come off for a while because it's going to be interacting with all the stuff. So first what's going to happen is you're going to get this void. And so the void is basically anything that is going, that like got stuck, that just like went straight around the beads. And so this is typically like aggregated stuff. And then so typically we set our fraction collector. So these lines are showing like the elution fractions. So like A1, 2, 3, 4. Those are like the different like wells in the 96 well plate that we set up. And so we actually set it up so that it's not even collecting the void. Sometimes you do want to collect the void if it's like a new protein or something, and you want to save it if it's aggregated still. But then we can start collecting the protein, and then you'll see that the bigger proteins are going to come off first, the smaller proteins are going to come off later, and then you can combine the fractions that you think have your proteins. So you combine like these peaks, and then depending on like how worried you are about this contaminating protein, you can make the peak like smaller. You can like use some of your protein yield, like potentially a purer sample. And you can run those on a band. So they're also, this was talking about more like purifying, so like preparative size exclusion chromatography, where we're basically want to make the protein pure. But we can also use analytical size exclusion chromatography, where we want to get a sense about the protein size and whether they interact, because if proteins interact together, then they're going to be bigger than they were individually. So they're going to run differently, and more on that in my other post. As to the term chromatography, it actually means writing in color, which I think is gorgeous poetically. And it comes because people were originally using chromatography to separate pigments. And so you might have done something similar where or purposely or not purposely, where you stick a mark, like different colored markers, and then you put it in some sort of solvent, some liquid, and then the dyes in the marker are going to separate as they go up. As they travel up, they get wicked up through like a paper towel or some sort of paper that the liquid is holding the pigment is going to get sucked through. And the basic idea of chromatography is that you have a mobile phase, and a stationary phase, and they're going to interact with one another. And basically the thing that was in your mobile phase gets to decide, okay, do I like the mobile phase better, or do I like the stationary phase better? So it can be a piece of paper and the liquid. So you'd have the paper would be the stationary and the liquid would be the mobile. Here we have our mobile is the liquid with our protein, so like our buffer containing our protein, and then the stationary would be the resin, the column. And so basically we're giving the protein the choice, okay, do you like the resin better, or do you like the liquid better? And so we're therefore able to separate based on how they're interacting with one another. And based on their differential interaction with the liquid versus the stationary, we can then separate things out that way. Hence, we call it chromatography. But there are other types of chromatography in addition to this column chromatography we use for purification. There's also gas chromatography, various forms of chromatography, thin layer chromatography, all these different things that you'll see in different fields like chemistry and that sort of thing. But for protein chromatography, we do it in these columns like this, and it is a super duper fundamental go-to tool for protein biochemists, and I love it.